everyone, and welcome to the second of our 6 Cafe Sci Talks this summer. Before I get started, as a nonprofit research institute, the discoveries and the solutions and the inspiration we try to create in the world are made possible with the help of our generous supporters. And I want to start by saying thank you to all of our donors, our board members, and our advisory board members. And as board chair, Bill Burgess likes to say, thank you for your time and your talent. We are very grateful and need it. And I especially want to thank H.M. Payson, who has been such a wonderful partner over the years with the Cafe Sci series. H.M. Payson is a Maine-based financial advisor who is sponsoring our Cafe Sci series again this year. So just a reminder, so please use the question and answer button at the bottom of your screen to send us questions throughout the talk. We're going to have question and answer periods twice, once midway through and then at the end. You'll also notice that there's a raise hand and chat button at the bottom, but those features aren't available in webinar mode. So you're not being ignored, they just aren't connected. So now it's my pleasure to introduce this evening's speaker, Dr. Kath Mitchell. Kath received her PhD at the University of Strathclyde, which is in Scotland. She is a physicist by training. And one thing about Bigelow is we really like to bring together scientists from a lot of different fields, and Kath is really a great example of that. And in true Bigelow tradition, she is bringing a fresh approach to the field of satellite oceanography. Kath joined Bigelow 6 years ago. She came to us as a postdoc working with senior research scientist Barney Balch. And this past year, I'm very excited to say that we held a national search where we had 113 applicants for new senior research scientist position. And Kath was one of the people we selected. We offered her the job, and I'm very grateful to say she accepted it. So our senior research scientists have to raise most of their own salary each year with contracts and grants, and Kath has really knocked that out of the park in this respect. She is currently leading or participating in a number of projects, some of which you'll hear about today. These include studying ocean color in the coast of the Gulf of Maine, a project involving mapping seaweed distributions along the coast using drones, and one on the impact of volcanic ash on phytoplankton in the Northeast Pacific. So, and one new project that she has that we're really proud of is she is looking at ways to detect swarms of copepods, which are the tiny kind of insect-like organisms that eat phytoplankton. If you've come to any of the talks by David Fields, he's the copepod fanatic. So she is looking at detecting swarms of copepods in the North Atlantic using satellites. And it's a really exciting project because this is funded by NASA through their new investigator program. So it's a prestigious program that really shows the confidence that NASA has in Kath as becoming a real leader within satellite oceanography in the coming years. And we agree she's fantastic. So with that, I give you Dr. Kath Mitchell.
Great, thanks Debbie, and hi everyone. Thanks for coming. I'm excited to talk to you all today. So, oh, sorry, slides. Okay, so as a kid, I was always fascinated with outer space. I love to look up at the night sky and look at the stars. And it was maybe a natural progression that I went out to study physics at university. This screen has just completely gone blank on me. I can't see anything. And so, do you want me to keep going?
That screen should not be right.
I don't know. I got logged out.
Kevin's
Okay, wait, it's back. It's back.
Okay.
It's back. Okay, sorry for the interruption, everyone. Yeah, so from this fascination of stars as a kid, I went on to study physics at university. And I learned there a lot about how scientists around the world explore outer space and how space agencies around the world build these really cool instruments that they launch out into the solar system and send to other planets. And, you know, maybe this part isn't a surprise, but despite that early interest in space, I didn't become an astrophysicist or an astronaut. I'm here talking to you today because I study the oceans, but I still get that kind of space kick because I study the oceans from space. So NASA and its counterparts around the world don't only focus on outer space and the rest of the universe, they also explore our planet. They have a whole bunch of these different instruments and satellites that look at the Earth and its environment and the ecosystem. The extent of this Earth observation fleet was pretty new to me when I started at graduate school, actually. And that's something coming from a kid who was fascinated with space and who studied physics. I thought we just had a lot of weather satellites and some fancy cameras that spied on you, but there's a whole load of different satellites up there with different instruments that all measure different parts of our planet. So the information that they can give us is pretty incredible. What you're looking at is an image that's known as the blue marble. And it was made by combining data and information from a lot of different satellites. And so these were, satellites that were designed to specifically look at the land or at the atmosphere and the clouds or the ice and the oceans. And this overall effect is this cloudy blue marble. And why does this marble look blue? It's not a trick question. It looks blue because the oceans are blue and they cover the majority of our planet. But if you look closely at the oceans in that image, what you see is they aren't all one uniform shade of blue. There's that dark blue in the middle, that's kind of in the middle of the Atlantic Ocean, but then there's lots of milkier blues or turquoises that are in other parts of the globe. And so what is going on here? Why are there all these different colors? And do these colors even tell us anything about what's living in the ocean or something about the ecosystem? And well, spoiler alert for the rest of the talk, yes, it does. And this is where my research is focused, interpreting these patterns in ocean color. So let's zoom in for a moment onto two different sides of the Gulf of Maine. What you're looking at is Cape Cod in the left-hand image and the Bay of Fundy next to Nova Scotia in the right-hand image. And what you can see in these really zoomed in images is a lot of detail in these different colors, and you can kind of get a feel for what all these different colors are. So, what are these colors actually telling us? Well, one of the things I love about satellite ocean color science is the origin of that question. And BIGO plays a big part in that story. Charlie Yench was one of BIGO's co-founders, and he wrote a paper in 1960 looking at the effect phytoplankton have on, and I quote, the color of seawater. And so when I say phytoplankton, I'm talking about the algae that are at the bottom of the marine food web. And what Charlie and his colleagues realized was the effect that these phytoplankton had on the color of seawater could maybe be visible from space. So they got involved in a project that was called the Apollo Suez Test Project, which in itself was pretty exciting. It, in 1975, became the first international effort of manned spacecrafts from two nations meeting in space. And those two nations were the US and the USSR, who the previous decade had been battling in this race for space. But they got together to try and meet their spacecrafts out in space and do some science together. And so the Apollo astronauts had been briefed to look out for changes in ocean color. And one of my favorite papers from Charlie was where he describes these observations. One of the science questions in that paper was simply, do astronauts see color changes in the oceans? And his conclusion was, the answer to this question is now obvious. Astronauts do see ocean discolorations. So off the back of what the Apollo astronauts saw and the work of Charlie and some others, NASA were convinced to launch a satellite dedicated to look at ocean color. In 1978, the first ocean color satellite was launched, and the images that came back were pretty incredible. There was all this detail and these patterns and these different colors that kind of hadn't really been fully imagined. And this satellite collected data for 8 years, and the ocean color community really grew. And we suddenly had all this information, all this data, but we didn't fully know what it all meant or how to interpret it. And the field was almost playing catch up. The theory and the methods were developed off the back of what some of these satellite observations were showing. And so the images you're looking at right now are not from that original ocean color satellite, but one that's operational today, Landsat 8. It's one of a series of next generation sensors that are used to look at the ocean. One of the things about Landsat, though, was it was actually designed to look at the land, and people have started to use it to look at the oceans. And so it's not perfect for the ocean, but what's special about it is how fine scale that information we can get is. So what you're looking at are patterns and changes in ocean color that are tens of meters in size. The original and most other ocean color satellites today can't resolve anything smaller than a kilometer, so 1,000 meters in size. And to give you an idea of what that means, with Landsat, we go from something like, if you look at the image of Cape Cod on the left, we're going something like that with all that detail to something that looks a bit pixelated and more fuzzy. So you're still seeing some general patterns, but you don't get that same fine scale information. But back to that initial question, what do these colors actually tell us? Whether they're at this high level of detail or not. And so the first thing that we get from ocean color is the material that's in the water. So these deep blues that are in the corner of this image, and what kind of covered a lot of the planet in that blue marble image that I showed before, that's pure water. If water doesn't have anything in it, it's pure blue. But when you start to add in other materials, then the color will change. So the big thing that ocean color scientists are typically interested in is phytoplankton. And so they color the water these different shades of green typically. And you can think of this like a pond full of algae, which I'm sure you've seen. You get this these green mats. And so a similar effect is happening in the oceans, maybe not quite to the same extent as in your pond, but the color change is still enough that we can see it from satellite. And then if we move over onto the Bay of Fundy image on the right-hand side there, within the bay, you have these lighter blues, almost like slightly brown colors. And what they're telling us is that there's a lot of suspended sediments in the water there. And so the tides, you have these big high tides in the Bay of Fundy. And so as that water is coming in and out, it kind of lifts sediment up and down off of the seafloor and mixes it up through the water column. And so you can think of it as just almost like muddy water. And that's kind of what you're seeing there. And the final material that we think about when we're thinking about ocean color is this browner stuff, which you see here coming in from the rivers. And we call that color dissolved organic matter. But you can just think of that as tea. So tea is just decayed plant material and dissolved plant material in water. And so that's exactly what you're seeing coming out of the rivers there. And that tea-like material comes from land plants that seep into the waterways and into the river, into the rivers and out into the ocean. But it also, you can get that similar material from the marine life that's in the ocean as well. Okay, great. So we have this satellite, we have this image of the ocean, we have all these different colors. We say, oh, look, there's a green patch. That must be phytoplankton. We're all done, right? Well, it's not quite as simple as that, because what happens when you get an area of yellowy green water, something like this closer in to Cape Cod? Well, what is that? Is that a particular type of phytoplankton? Is that a mixture of phytoplankton and some of this tea material, or some muddy water as well, or a combination of all three? How do we solve that problem? Okay, so this problem is kind of the classic remote sensing problem. And we call it the inverse problem. It's relatively easy to go in the forward direction. So by that, I mean, we have a jar of phytoplankton, and we have a jar of tea and a jar of muddy water. And then I mix it all together and I end up with something that's this browny green color. So ultimately I can say I've got this much of each of these things and it gives me this overall color of whatever. But if I was to go out on a walk and I passed a river or a lake, or I was at the coast and I scooped up some water in a jar and I looked at its color, it's something like this. Can I go in that backwards or inverse direction and say, like, there must be this much phytoplankton and this much tea and this much mud that's in the water? And that's actually a big challenge. And that's what I spend my time doing. Okay, so why bother? Okay, great, you can tell what's there. How is that useful? Well, you can tell a lot more than just what's there. You can tell how much there is. And that's what's really important for us understanding life in the oceans. And so phytoplankton, these are the algae, they're at the base of the marine food web. And they support ultimately all life in the oceans, be that the zooplankton that directly eat the phytoplankton, or the larger mammals like fish or whales, or even seabirds that eat the zooplankton or eat the fish, or somewhere in between. And so if we know where the phytoplankton are and when they are there, it can help us understand the habitats for all these kind of higher level species. But phytoplankton also play a major role in regulating the climate. So the Earth system is all connected. You can't really look at one part in isolation from another. Phytoplankton use carbon dioxide, drawing it from the atmosphere into the ocean and ultimately into their cells. And so as well as removing that carbon dioxide from the atmosphere, phytoplankton produce oxygen and release that into the air. In fact, phytoplankton and bacteria in the ocean are responsible for producing 50 to 80% of the oxygen that's on the planet. And so when these phytoplankton die, they begin to sink down to the seafloor. And so they actually take some of that carbon that they'd removed from the atmosphere and have in their cells, they take that down to the seafloor with them. And so phytoplankton are playing this pretty important role where they're moving carbon around the planet and in the oceans, and studying them not only tells us about marine life, but also about the cycling of carbon, which is linked to our climate. And so the great thing about using satellites to look at ocean color and to look at phytoplankton is you can measure how much phytoplankton there are over the whole globe on a daily basis. So what you're looking at now is a video that's called The Earth's Biosphere, and it's made from 20 years of satellite data. And there's changes that are happening in the ocean and on the land, and we're just going to focus in on the ocean right now. And what those ocean colors are representing is how much phytoplankton are there. Sort of the phytoplankton biomass is typically how we refer to it. And so the darker purples and blue colors are where there's not as much phytoplankton, and the greens and yellows and reds are where there are a lot of phytoplankton, or at least the most phytoplankton. And so what you'll see is the planet is spinning, is time ticking on throughout the year on year. And you'll see there are some fairly consistent patterns there, like these bigger areas where it's typically always low phytoplankton. But then you'll see these changes that happen at the poles and along the equator. And these are like seasonal changes as phytoplankton are growing in the spring in the northern hemisphere and over the summer, and then declining over the winter as it gets darker again. And you know, a lot of people that have described this video describe this as like a beating heart for the planet, or the earth breathing, and that's being really driven by these phytoplankton that are at the base of the marine food web. So if we think about what we're looking at here, we have this continuous data stream of our ocean ecosystem from 1997 until today. That's 24 years. And so this helps us move past these annual changes that we're seeing there to look at longer term changes that have happened over that full data record. And we're only looking at one thing in this video, phytoplankton biomass. There's really a lot of other things you can measure from ocean color as well. And that's where my research really comes in. Coming up with new methods to measure new and different things from ocean color. And so how do I do this? So typically I'll work with other people to go out on a ship. We'll make some measurements of ocean color, similar to what a satellite is making, and whatever other thing it is we're interested in that, whether that's how much carbon there is, how much phytoplankton there are, and as I'll talk about later, how much copepods there are. And so we go out, we do these measurements on the ship. And then, you know, I'll also do some measurements in a lab where I ultimately look at how different materials will affect the overall color of the water. And then I spend some time sitting at my computer doing some modeling, making some predictions about how these different quantities of different materials ultimately affect the color. And then I'll apply what I learn to satellite and look at that long-term record and see what patterns we see and see what changes we see. But what's important is all these steps are not done in isolation from each other. They're all connected. Every different part informs every other part. And what you really need this integrated approach to pull together the different bits of information and to create some new understanding. This brings its own challenges because you have to try and combine all the data from all these different approaches. And I'd like to come back to something I said earlier. We've had an almost daily view of the ocean for the past 24 years, and it's continuing into the future. So that's a lot of information, and that's ultimately a lot of data to handle. And so the types of instruments that we now use on ships are the new type of autonomous robots that we send out into the ocean. They collect a lot of data also. So you're not only connecting lots of different types of information, but you're connecting a lot of it. And so to do this type of work, I need to write my own computer programs to work with the data and develop the methods. This isn't something that I can do in a program like Microsoft Excel. And these kind of skills or this kind of approach to working with oceanography or oceanographic data is becoming more common, but these skills haven't really been traditionally taught as part of undergraduate oceanography degrees. Here at Bigelow, we're building out some of our education programs to teach some of these skills. And something that I'm really excited about is an event that I'm helping to host here in August called Ocean Hack Week. And it's a workshop where we teach some of these computational skills, but a big part of it is the participants will then split up into small groups and work together on some oceanographic project that one of them or them as a group have come up with that they want to work on. And so while we're only hosting people from the US here on site because of the pandemic, there's a virtual component too, and that involves people from all over the world. And so it's a really great event to learn some new skills, but to build some of these international collaborations as well. So with that, I'm going to pause for a break and pass it over to Debbie to moderate some questions.
Oh. Okay, so let's see. First question, do clouds or the atmosphere cause issues when using satellites to look at ocean color?
Yes. Yes is the answer to that question. Both cause different problems. So when I'm talking about color, I'm talking about visible light. So that's something that you can see. So if you imagine you look up, you can't see through the clouds. It's the same for the satellite. They can't see through the clouds from above. So if there are clouds in the way, we can't see the oceans. And that was actually a problem on that Apollo Suez test project that Charlie Yench was involved in, because Charlie was actually on a boat in the Gulf of Maine and knew there was a phytoplankton bloom happening. And they relayed that information to the astronauts, and they looked, but they said, it's cloudy, we can't see anything. So it was pretty fascinating. They managed to do that, but unfortunately no data. And then the atmosphere itself, because the light comes from the ocean and then goes all the way to the atmosphere until it's seen by the satellite. And so we have to remove that atmospheric component. And that's a whole other science in itself that people spend their lives doing. So yes.
Excellent. You talked about using the land satellite for looking at the oceans to be able to see things in more detail. Why haven't ocean color satellites been designed to measure the oceans in that high detail?
So the thing that's going on that we have to balance is how much detail do we want on a spatial scale? So like how small can we see things versus like how frequently do we want to see things? So the way that the ocean color satellites have been designed are to look at the oceans, which are very large, and to try and get a daily view of the whole planet. So what that means is, the satellite actually orbits the Earth once every 90 minutes. And then it does that however many times to get the whole way around the Earth in one day. To be able to design an instrument that can look at the higher detail, you lose some of that temporal resolution. You don't get a daily image. So Landsat, for example, its revisit time, I think we get an image every 1 to 2 weeks. It's just one shot of part of the Gulf of Maine. And so if that 1 to 2 weeks later there's clouds, then you're unlucky. So it depends what you're looking at, what instrument you ultimately would end up using.
Okay. You said the land satellite wasn't perfect enough, though it gave you detailed images. What's wrong with it?
So it was designed to look at the land. And so ultimately, what your eye is seeing is like every wave, every color of light that's possible that you're in the what we call the visible spectrum. But these satellites only look at a couple of wavelengths of light, so a couple of colors. So imagine I have a green view of the world, and then a blue view of the world, and a red view of the world, and then I try and piece everything in between together from those 3 slices. Landsat only has, I don't remember off the top of my head, but 3 or 4 slices or different colors, whereas the ocean color ones have a few more. And the ones that the ocean color sensors have are the colors of light that are particularly relevant for looking at phytoplankton. So that's kind of the big difference there, is that the Landsat satellites were designed to look at the land, but we're doing our best to use them to look at the ocean. Okay,
are you looking at calcite aragonite and the effect on ocean acidification?
Why I first came to Bigelow to work for Barney Balch was to work on methods to look at calcite from satellite. And so Barney's done a lot of work in that field. I've also done a lot of that work now, and we're continuing working on that together. And so we're looking at, we have methods for detecting calcite from satellite, and that's really driven by a particular type of phytoplankton, coccolithophores, that have these calcite shells. But we are not, from satellite, looking at those other forms like aragonite. And as to the question about the effects that has on ocean acidification, that's actually a big part of this project, one of the NASA projects that Debbie mentioned that I'm funded on, is to really go back through our satellite data record and see what changes that we've observed, both in the satellite and in the long-term time series data that we have in the Gulf of Maine. So I don't have answers to that question yet, but it's something that we're looking at.
Okay, and last question for this break, and there's more, and I'll ask them at the end, but how do plastics affect color?
That's a good question, and it kind of leads into something that is tangential to what I'll talk about later. But it depends on the size. So if you can have microplastics that are suspended and they're really small, and then you can have like these bigger mats of floating plastics. And so these bigger, there's been quite a bit of work done in the last few years looking at these larger floating mats of plastics and using those kind of high resolution satellites as well, things like Landsat, because these kind of, these big mats of, you find that like plastic bottles and fishing nets or whatever kind of materials will stick together and float around in the ocean. And so people are working on methods to detect those, and they're kind of promising. But the microplastics, that's an unknown. There's a lot of ongoing research. I'm not directly involved in that people are doing to look at whether we can see effects of those smaller scale plastics on the overall ocean color.
Okay, so I'm going to save the rest of the questions to the end, and so you can finish the talk. Thanks, Kath.
Great. Okay, so for the second half, I want to talk to you about a couple of my research projects. And these projects are all very new and only just really taking off. So I don't have any science results to share with you, but I just wanted to share with you some of the things that I've been talking about and how I'm pulling together all these different strands that I was speaking about before break. So I'll start by putting these projects in a little bit of context. So I spent a long time talking about phytoplankton in particular. And when ocean color remote sensing took off, it was really focused on using satellites to look at phytoplankton. And the first project that I'm going to talk about is to do with phytoplankton, but I've started to think about using satellites and remote sensing for looking at some larger things. First off, the zooplankton. And so these were the critters that eat the phytoplankton. And in particular, I'm going to be looking at a type of zooplankton copepod called Calanus. And so these zooplankton are the ones that are eaten by the North Atlantic right whale. And then the second thing that I'm going to talk about is I'm looking at seaweeds. And these are seaweeds that grow along the coast in Maine, in the intertidal zone. So that's the part that gets covered by high tide, but is exposed by low tide. So these are seaweeds that sometimes are underwater and then other times are not underwater. And so let's start by focusing in on the Phytoplankton Project. And so this is a project that was recently funded through the North Pacific Research Board. And here I'm working with a team of people to look at the effect of volcanic ash on phytoplankton blooms in the North Pacific. And so the North Pacific is this region of high nutrients, but low phytoplankton biomass. So there's not that much phytoplankton there. And one of the reasons for this low phytoplankton biomass is the lack of available iron, which is something that is also important for phytoplankton to grow. But there's been evidence that after volcanic eruptions, there are these large phytoplankton blooms. So this period of increased growth, with the thought that it's the volcanic ash that's providing the iron to the phytoplankton. And so a relatively newly identified potential source of iron is remobilized ash. And so this is ash that, if you imagine an eruption, and you have this fresh ash that goes up into the atmosphere and comes out and falls on the ocean. The remobilized ash is ash that's after the eruption is fallen on the side of the volcano or on the land nearby, and it lies there for a while, and it weathers and it ages, and then there's a big windstorm comes along, and then it picks up that aged ash, it remobilizes it, and then it gets blown out and falls on the ocean. And so a colleague of mine up at Colby College here in Maine is Bess Kaufman. She found that this remobilized, this aged ash contains iron in a form that's more useful for phytoplankton to grow. So Bess and a couple of colleagues here at BIGL, Ben Twining and Karen Stamieskin, and I are going to investigate the impact that this remobilized aged ash has on phytoplankton growth in this North Pacific region. And what you're looking at is a schematic that Karen put together that highlights all the different aspects of this project. And you can see that it clearly demonstrates this idea of an integrated approach that I was talking about before. So Bess and Ben will do some of the lab geochemical analysis of the different ash samples from fresh and aged ash. All of the team at Bigelow will be involved in a couple of cruises where we do some shipboard experiments where we'll add the ash and measure the effect on phytoplankton growth. And then I'll be doing some lab work to measure the optical properties of ash and see how it will affect the overall ocean color, because the presence of ash in the water or atmosphere will affect the common methods that we use to detect how much phytoplankton are in the water. And what we ultimately want to do is look at the long-term satellite record to identify these remobilization events, because you can see that in the satellite data, and then connect these events with potential changes in how much phytoplankton there is in the water after those events from the ocean color signal. And the final aspect of this project, as well as this Western science approach, is we're going to be working with the Aleut community on the Pribilof island of St. Paul, which is in the middle of the Bering Sea, which is right here in between Alaska and Russia. And what we're going to work with that community to integrate their traditional knowledge of volcanoes and the effect on ecosystems with our findings. And we're going to spend some time each year on the islands with the community and work with them and learn from them. The Aleut people are indigenous to the Aleutian Islands and rely on surrounding waters for the fish, the seals, and other sea mammals. And as I spoke about earlier, phytoplankton support the full ocean ecosystem. So by learning more about what's influencing the phytoplankton growth in the waters surrounding the Aleutian Islands, we can learn more about those habitats for those higher trophic level species, for those fish and the seals, which are ultimately what the Aleut rely on. Okay, so we'll now move on to remote sensing of zooplankton. So here, I'm going to talk about using satellites to look at the copepod Calanus. And I'm going to be looking at a couple of different types of Calanus, which are found here in the Gulf of Maine and further up the coast into the Gulf of St. Lawrence. And Calanus are a really important player here in the food web, in the marine food web. They are the prey of the critically endangered North Atlantic right whale. And so the North Atlantic right whale have been observed to eat Calanus when they swarm. So when you suddenly get a lot of Calanus in the water, that's when it's most advantageous energy-wise for a whale to just open its mouth and get as much copepods as it possibly can. And then the thing with Calanus is they have this red pigment in them. And there's been reports from as far back as the early 1900s stating that the water looks red when they swarm. So if you can see that color change with your eye, then you should be able to see it from space if the swarm is large enough. And this does seem to be true. What you're looking at is an image from a recent study that showed it was possible to see Calanus when it swarmed off the coast of Norway. This work was done by Basedow et al. A couple of years ago. And to give you a bit of a sense of scale here, so phytoplankton are smaller than the width of one of your hairs, the hairs on your head. Calanus are 4 to 5 millimeters in size. So, you know, a thickness more of like 10 human hairs. It's still pretty small, but you can see it. And this swarm in the Norwegian waters there's a scale up there in the top corner. It was 100 kilometers across. So more like a couple hundred kilometers across. And so 100 kilometers that's about 60 miles. And so I've been funded by NASA to team up with some people who participated in this cruise to analyze some of the ocean color measurements that they made to really try and quantify their initial observation of how Calanus are influencing ocean color. I'm also working with other people here at Bigelow and in Canada to look at records of Calanus observations from around the North Atlantic, as well as that's from field data, as well as from satellite data, to see if we can identify any swarms from space in the Gulf of Maine and the Gulf of St. Lawrence. And we're aiming to kind of get to the point to say, here is a Calanus swarm, rather than there's this many Calanus in the water. And if this is successful, it would be really helpful for conservation efforts of the North Atlantic right whale. If you're local to Maine, you may have heard a bit in the news over the past few years about the decline in the population of the North Atlantic right whale, despite some conservation efforts that are in place. And there's been part of the problem has been that there's a lot of ship strikes and gear entanglements of whales. And there's been some recent evidence that a change in the foraging behavior of the whales suggests they've moved out of the Gulf of Maine, where they used to always be, to follow their prey, as their prey has moved because the Gulf of Maine has got warmer. And so this movement has meant the whales are in regions with less restrictions and less conservations in place. And so there's a larger possibility of ship strikes or gear entanglements. And so if we have a method that we can ultimately detect copepod swarms from space, it means we can use the satellite data to show where the whales are more likely to be, because we know that they go to these swarms to feed. So with that long-term field data record and satellite data record, we will be able to see if these swarms are indeed moving up further north from the Gulf of Maine and up into the Gulf of St. Lawrence. And so the final project I want to talk to you about involves macroalgae or seaweed. So we've gone from phytoplankton, very small, to copepods, to slightly bigger, to seaweed, which is much bigger. And so for this project, I'm working with a team of people to develop methods to use drones to classify, map, and estimate biomass, so how much there is of two common seaweeds here in Maine, Ascophyllum nodosum, which is commonly known as rockweed, and Fucus vesiculosus, which is commonly known as bladderwrack. And both of these seaweeds are ones that are found in this intertidal zone, so that they're spend part of their life fully submerged by water and part of their life exposed. And so this project is a bit different from me. For me, the drone is effectively measuring color. So even though the system is very different from phytoplankton floating in the ocean, the types of measurements and the interpretation is very similar to the ocean. I've never worked with seaweed before, so it's been a very steep learning curve on that side of things, but I'm working with a great group of people that have this wide range of backgrounds. And this project was funded through Maine Economic Improvement Fund, Small Campus Initiative, and Biggles Centre for Seafood Solutions. And so on the team, there's the seaweed experts from Maine Maritime Academy and Schoodic Institute, both further up the coast in Maine, as well as Nicole Price and Brittany Honish here at Bigelow. And then there's the drone expert, who you're looking at, Stefan from Nearview LLC. And then the fun part for me is there's a land-based remote sensing person, so someone that has done a lot of research using remote looking at land from drones or from satellite. And then there's me, the ocean person, that's done all this work looking at the oceans from satellite. And we're trying to come together to look at this zone that's right in between the two. And so there's a few different aspects to this project. There's this growing industry around seaweed harvest, and there's been really no surveys to quantify how much of these common seaweeds there are along Maine's coast. So to ensure a sustainable harvest, we need to know what is there and how much is there in the first place. So we're all working on methods to map and classify the rockweed and bladderwrack, and that's going pretty good. Like, we can kind of tell them apart. But then this other aspect is estimating biomass, how much seaweed is there from the signal that the drone is collecting. And so for this part, I've had a student working with me this summer, Lydia Duncan from the University of South Carolina, and she's been investigating whether or not we can estimate how much seaweed there is based on what the drone can see. So to do this, we joined the rest of the team out in the field for a 2-day sampling session. And in these sessions, on the first day, the drone flies overhead and maps the full site. And then we do some ground measurements of the color as well with an instrument similar to the one that's on the drone. So we can basically ground truth the drone. And that's what Lydia is doing in that picture that you're looking at. She's got the instrument on her back and she's got the sensor, the detector part in her hand that she's pointing down at the seaweed. So then on the second day, after the drone part has been done, we, at 50 locations across the site, we put out these small square quadrats. So that's that white thing that Lydia is holding in her other hand. We put that down, and where that is, we measure the color with that handheld instrument again. But we also measure the depth, or the thickness of the seaweed bed, how much seaweed is there. And we also look at how much, we'll weigh how much rockweed is there and how much bladderwrack is there. And we'll combine all these data to investigate if we can measure biomass from the color. So as well as those days in the field, we spend a day in the lab making very controlled measurements to get a handle on some of the variability that some of the changes that we see just from the nature of doing this work out in the field. For example, changing light conditions will really affect the overall color. And this is easiest to imagine if you think about having a red light bulb. And then everything in your room changes to having a very red hue, a very red color. And so this same effect happens if the sun goes behind a cloud. And it's maybe not that noticeable to the human eye, but it is to the instrument that we use. It's sensitive enough that those small changes in light conditions will affect the reading that the sensor gets. The other fact, so we set up this, almost looks like a photo studio in the lab where we have these lights all set up, and we leave it at that constant level over the course of the day. So we can move our samples in and out and measure the color of those different samples. But the other aspect that's really hard to control in the field is how wet the seaweed is. And so as it dries, the color changes a bit. So we want to be able to control for that too, because ultimately what that means is if the tide has just receded and the seaweed that's exposed is still very wet, if that the drone flies over at that point and the signal is very different than if it flew over, 6 hours later before just as the tide's coming back or before the tide comes back in, and we get different results, but the seaweed hasn't changed. So we need to kind of get a handle on whether the time that if the seaweed has dried out at all, if that's going to affect our data. And that, again, is something that we can do really easily in the lab, where we can just repeatedly measure that same, you'll see that seaweed in that picture, it's on a piece of card. So, we can repeatedly measure that sample over the course of the day. So that's what we did early in the summer. And Lydia has been working really hard to process all that data in the lab. And she's just in the final weeks of her project. She's going to be presenting her results here at Big O next week. And those results are really promising. It seems like there is a relationship between the color and how much seaweed there is at lower biomasses, but that relationship breaks down as the thickness of the seaweed increases. But this is kind of what we suspected, because the instrument's ultimately just kind of seeing this top layer. And so once the thickness of the seaweed bed gets too much and the instrument's only seeing this top layer, it doesn't really matter what's below, because it's not being detected by the instrument. But we were trying to see if we could get a handle on how thick that cutoff is. And so it looks like that's happening, but really we need some more data to nail that down and make that relationship a bit more robust. But that's great, because actually today and tomorrow, the rest of the team are out in the field over at Pemaquid, so just across the river from where I am right now at Big O. And I'm going to go out and meet them tomorrow and collect some more seaweed and bring it back here and do some more measurements in the lab on Thursday to get some more data for this project. And ultimately, if we can come up with this robust method, what this means is we can use drones to survey these coastal zones in these seaweeds. And these methods could even be scaled up to satellite. So you could classify and map the intertidal zone over the whole coast of Maine, and learn the biomass of the seaweeds that are there. And these data can then ultimately be used to inform harvesting of seaweeds. There were these were three examples of projects that I have had in the pipeline and are kind of in their early stages. And I'm really excited to start working on these projects properly and to really try and push the boundaries of ocean color remote sensing to encompass these kind of new areas as well. But I'm also excited to continue working at Bigelow to expand our education programs and to teach some of these computational skills that I use to people that need them, that really need them to work in these kind of information-rich or data-rich sides of oceanography. And so I just want to end by thanking my funding agencies and acknowledging the contributions from colleagues and collaborators and students that kind of went into the content of this talk. And before I stop for questions, what you see there is a quote, and I'll read it out to you in a minute, so you don't have to try and read it while I'm talking to you. But this is a quote from Charlie Inch. And so Barney Balch, who was the senior research scientist that I came here to work for, when I first moved over 6 years ago, had worked at Bigelow himself on and off since he was in high school. And he worked closely with Charlie. And Barney has this quote pinned up on his office wall, and I finally took the time to read it last week. And Barney told me this is something Charlie had written in one of his annual reports he'd written to NASA when updating them on the work he'd done for the year. So we as scientists who get funding from external sources have to write these annual reports to update our progress. And this is not normally the type of language that I write my annual reports in, but maybe I should take a leaf out of Charlie's book. And so this is what Charlie said. Without global ocean color satellite data, humanity loses its capacity to take Earth's pulse and to explore its unseen world. The fragility of the living Earth and its oceans has been noted by astronaut Sally Ride, who speaks of the changes in land, oceans, and atmosphere systems as witnessed from space, and the challenge that a changing climate presents to our beloved Earth. It is our duty to provide a long-term surveillance system for the Earth, not only to understand and monitor the Earth's changing climate, but to enable the next generations of students to make new discoveries in our ocean gardens, as well as explore similar features on other planets. Thank you, and I'll pass back to Debbie to do more questions.
Excellent. Thank you. Thank you. Thank you. So we've got lots of questions. See, this is Richard Betts. I'm sorry, I didn't say who was asking the questions before. Are drones playing an increasingly larger role in your studies? Which you just described that. So shoot, I didn't read that all the way through. Well, I'll expand that a little bit. So you're definitely doing drone studies. How common is that becoming?
It's a good question in terms of studies in the ocean. So it depends on who the drone owner is and the drone pilot is, because drones are really expensive and not everyone wants to fly their drone out over the ocean, because if something goes wrong and then it falls in the ocean, it's really hard to get it back. But that being said, more people are starting to use it to look at these smaller water bodies, so inland lakes, or very close to shore, at the coasts, because satellites can't really see the detail like in the Damariscotta River out here. And so it's becoming more common. People have been looking at drones of various different types for a while. It's really challenging with the ocean, because the thing with the ocean is the ocean surface changes, and that really affects the ocean color ultimately that the drone is measuring. But there is definitely a push towards that for using it over open water as well. But those methods are just kind of developing right now.
Okay, this one is from Charles Maisel. Are you seeing long-term trends in phytoplankton distribution that can be interpreted in light of climate change?
So that's not something that I am directly looking at myself. A lot of, or most of my research is really kind of developing the methods for other people to go and answer those questions. I can speak a little bit to the Gulf of Maine because I'm also part of a long-term time series. If some of you are frequent listeners to these talks, you'll probably heard Barney in the past talk about NATS, the Gulf of Maine North Atlantic time series. And so there's definitely been changes in phytoplankton in the Gulf of Maine over the last 20 years as part of that, that we've seen in those field data. And, you know, as part of that, we're also seeing these warming temperatures in the Gulf of Maine as part of climate change. And so, so yes, we're seeing some changes there. But I am not looking at that question directly.
Okay, so this is from Susan Burnett. As the interactive global map turned, I noticed, and this would have been the first half of your talk, because this is a question from the first half. I noticed some coastal areas in red. What does the red color signify? And she follows it up with, so perhaps the fringing coastal red areas and the spinning interactive globe signify seaweed.
And so it actually, and that's a really good question, it actually signifies probably some, so there's a big one that you would have seen that comes out from the Amazon River plume. And so, you know, some of that is real information, you know, chlorophyll content, phytoplankton concentration. You get more phytoplankton in near the coast because the sea is shallower, and so there's kind of a bit more cycling round of some nutrients that the phytoplankton need to grow. But also those standard methods that we use to interpret the ocean color data into phytoplankton, they get confused when there's this sediment or this mud in the ocean as well. So some of that really high red signal around the coast is actually maybe slightly wrong, slightly elevated, because it's actually some sediments that are in the water as well as the phytoplankton are there.
Excellent. Okay, Leanne Whitney. Leanne is one of our scientists. Great talk, Kath. Would you expect the season to impact your seaweed results? I'm thinking with regards to the length of the day, i.e. sunlight, with changing seasons and how that may impact properties of seaweed color, like fall leaves changing color.
Yeah, that's a great question, Leanne. So Leanne and I share an office, and she's sitting like directly above me right now. So we have squeezed all our sampling into the summer season for multiple reasons, part of which is we have this big cohort of students available to help us do all the field work. But something that we did do is we did one quick kind of recce day, like test day back in the spring when the seaweed was going through one of its reproductive cycles, because when the seaweed is reproductive, and this is like, you're pushing my limits of the seaweed here. When the seaweed is reproductive, it gets these little nodules on it that for the seaweed we're looking at, kind of orange in color. And so depending on like how reproductive it is, it does change color when it's going through that phase. So we went out and did some quick measurements then, but I think it would be good, and we've talked as a group about some of those seasonal differences that might happen. I'm not so sure about like the fall part myself, but I think it would be good to do some more in the spring and kind of through that reproductive season as well. But yes, it will change because of that.
Excellent. This is Peter Neidhart. How does your access to satellite data work? Is it public domain data, or do you subscribe to satellite feeds? How is it funded, and so forth?
Yeah, great question. And so all the satellite data that I use is publicly available. And so most of the data that I use come from NASA-based satellites and NASA assets. So, you know, they invest all this money into building these instruments and launching them and putting them up there. for us as scientists to use that data. And so NASA have this platform and this system that's made up this that you can go online, click a few buttons and get satellite images of wherever you want around the world. Or you can interact with it in different ways depending on how much data you want. And space agencies from around the world do similar things as well. There are some privately available satellite data. So some of like that really high resolution satellite data, looking at things in really fine detail, a lot of that stuff is available through commercial satellites that you would have to pay for. But I do not use that data. I just use what is there that's publicly available to us. And any new data that I create off of the back of my work, I make available to the public as well. Part of us being funded through government federal funding agencies is that, you know, they're giving us data, satellite data to use, and we give our results back out to the world again. And it's this idea of sharing all our science and all our data and all our methods with everyone that's really important to push science forward.
Excellent. And let's see, this will be the last question. Hopefully you are using VIRS data from NOAA as well as NASA data. Correct.
We are using, well, yes, I will be using the VIRS data from NOAA as well as from NASA. So VIRS is an ocean color satellite as well, and the data for it are managed through NOAA rather than through NASA. Or, well, there's an agreement between the two. But yes, VIRS is kind of the current ocean color satellite that's up there. The other ones are kind of in the very elderly stage of their life cycle. And so it's the data from VIRS that I'm kind of going to be using moving forward. And so that picture I actually showed you of the Calanus swarm off of Norway, that was from VIRS data, VIRS satellite data.
Excellent. Okay, so I'm going to close the Q&A. And thank you so much, Kath, for an excellent talk. Very informative. So next week, I want to encourage everybody to come back. Next week, we're going to hear from Maya Groner. Maya will be joining Bigelow as a senior research scientist in November this year. She's moving to us from Seattle. She will be joining us remotely to describe her work in Lobsters in a Pinch, Impacts of Warming Oceans on Epizootic Shell Disease. So with that, I want to thank all of you for coming and joining us tonight and for caring enough about the ocean and about Bigelow to learn what we're doing and how we're trying to protect the ocean. I see you all next week and that you have a lovely evening. Thanks. Bye.